Welcome back to the deep dive. Today we are digging into something fascinating under our feet, the secret life of plant roots. We know plants kind of feed their neighbors, right? This whole root microbiome, the rhizosphere, using these chemical signals, root exudates.
Yeah.
But the puzzle, the real head scratcher, has been how they manage where these microbes actually set up shop. It's not just random. And while new research suggests the community structure, it's really defined by these tiny controlled leaks. Surprising, isn't it?
It really is. That's our mission today, to get into the spatial side of things, how colonization actually happens on the root surface. And it turns out some of the busiest microbial spots aren't where the plant is actively putting out signals. Instead, it seems they pop up where a key internal plant barrier, well, where it fails, basically, creating these little temporary leaks of really energy-rich stuff.
Okay, so like a security system breach. When the security fails, bacteria just rush in. What is this security system, this gatekeeper you mentioned?
Yeah, exactly. It's a really critical layer of cells inside the root called the endodermis. Think of the root like a sophisticated filter. It needs to pull nutrients in from the soil, but in a controlled way. And it has to protect the core pipelines. It's called the stele, where water and nutrients travel up the plant. The endodermis is like the sealed border patrol right around that core.
Sealed border patrol.
Okay.
And what
Yeah.
makes it sealed? What's the actual seal made of?
That would be the Casparian strip, usually just called the CS. It's incredibly important. It's basically a band, a ring made of lignin, like woody material that gets impregnated into the cell walls. It sits between the endodermal cells and completely blocks the space between them. That space is called the apoplastic space. So it acts like, think of it like really good mortar between bricks. It stops any water or dissolved stuff from just seeping freely between the cells.
Right, forces everything through the bricks, the cells themselves. Why is sealing that space so vital?
Well, it does two key things. First, like you said, it forces everything wanting to get into the central plumbing, the stele, to actually pass through a living cell membrane. This gives the plant total control over uptake, selective absorption. Second, and this is super relevant for today's topic, it prevents the plant from just leaking valuable resources out. The stele is packed with sugars, amino acid stuff the plant worked really hard, energetically speaking, to make. If that Casparian strip barrier isn't complete, all that energy-rich goodness can just leak back out into the soil. It's a huge waste.
Ah, okay. Which leads us right back to the mystery the researchers were facing. They saw bacteria consistently gathering in areas where this Casparian strip was known to be patchy or broken. So if the barrier failed, something valuable must be leaking out, attracting them.
Right.
But what was the signal?
Exactly. That was the hypothesis. The defects must be releasing something specific, probably something highly concentrated right from the plant's internal transport system, the vasculature. The only way to figure it out was, well, metaphorically, to break the dam and see which river flows out most strongly.
Okay, let's talk experiments then. They looked at the stuff leaking out the exudates from mutant Arabidopsis plants. These plants, let's just call them the leaky barrier mutants, they have this known chronic problem with their Casparian strip being faulty. So what poured out when the dam broke?
What they found was a flood of amino acids. But what was really striking, shocking really, was the dominance of one particular amino acid, glutamine, or GNN for short. It made up over 80% of the total amino acids leaking from these mutant roots.
80%. Just glutamine. Why that one specifically? Not sugars or a mix of proteins or...
It comes down to simple logistics, really. Inside the plant, GNN is the main molecule used for transporting nitrogen around. It's the central currency for nitrogen delivery to the shoots. The plant actively pumps GLN into the xylem vessels. Those are the main water transport pipes deep inside the stele, behind the endodermis. So you picture the Casparian strip as that dam wall again. GLN is the big high-pressure river flowing right behind it. When you get a crack in the wall, it's that main highly concentrated river that's going to spray out most dramatically.
Okay, that makes a lot of sense. The leak isn't random. It's directly revealing what the plant is actively moving internally. And they didn't just rely on the mutants, did they? They actually proved this leak causes the bacterial pileup using a really neat, precise technique.
They did, yeah. They used something called localized laser ablation. Super precise. They took perfectly healthy, normal, wild-type roots and used a laser to basically zap tiny microscopic holes in just the endodermal cells. And bingo,
When they damaged the cells that form the Casparian strip, they saw bacteria accumulating massively and rapidly, like within minutes, right around that damaged spot. It
Yeah.
was pretty conclusive proof. The endodermis controls where these nutrient hotspots appear, and any breach acts like this incredibly attractive beacon for bacteria.
Wow. Okay, that's a huge piece of the puzzle. The endodermal breach is the signal flare, the GPS coordinate for the bacteria. But what happens once they get there? Is the glutamine just a really strong smell, or is it also, dinner?
It's absolutely both. And that dual role is critical for understanding how these microbial communities actually get established. The glutamine leak provides both a powerful chemotactic cue, that's the smell, the attractant, and it creates a rich metabolic niche, basically, the food source, that allows them to multiply, to proliferate.
Right, two functions. Let's break that down. First, the chemotaxis, the smell. How did they demonstrate the bacteria were actively swimming towards the lead, not just bumping into it by chance?
That's where they brought in the bacterial genetics. They used a well-studied model bacterium, Pseudomonas protogens, CHA0. First clue was speed. Like I said, the bacteria piled up around the leaky roots really fast, within minutes. That's faster than they can reproduce, so they must be moving towards it. Then they tested movement directly. They used mutant bacteria that were non-motile, basically. They couldn't swim. And guess what? Those strains couldn't colonize the roots effectively at all.
Okay, step one. Got to be able to swim. But
Yeah.
then, how do you prove they can smell the gluradan specifically, that they're navigating towards it?
Right. So they used two clever mutants for that. First, they had what you could call the non-sensing swimmer mutant. This one could swim just fine. Its motor worked. But it lacked the guidance system. It couldn't sense chemical gradients and change direction. This mutant, THK1 was its designation, was really bad at colonizing those known leakage hotspots, like where new lateral roots emerge. It could swim around, but it didn't know where the good stuff was.
So it's like they could hear the dinner bell faintly, but couldn't figure out which way the kitchen was.
Exactly like that. Then, to really nail Galen as the specific attractant, they made another specialized strain. This one you could call the Gillian-specific non-smeller. This mutant, Pictoquad, was missing just the four specific sensors, the chemoreceptors, known to detect amino acids. And sure enough, this strain completely lost its attraction to the Galen leaks. It wouldn't swim towards them, but crucially, it could still swim towards other things roots release, like citric acid. That really pinpointed Galen as the major, highly specific chemical signal defining these colonization hotspots.
Glenn is the siren's call. Got it. Okay, now for part two of the function, the meal. How did they show that glenn actually fuels bacterial growth once they arrive?
Yeah, the proliferation part, the evidence there was pretty clear too. They looked at bacteria already sitting on the roots. On the chronically leaky mutant roots, the bacteria multiplied about 3 times faster over a 3-hour period compared to bacteria on normal wild-type roots. And they did this cool experiment, kind of like a leaky bathtub water test, you could say.
Okay, they took the water or the growth medium that the leaky roots had been soaking in.
Exactly. They collected that medium, which was now full of the leaked glenn and other stuff, and then they put perfectly healthy normal roots into it. And even on those normal roots, the bacteria suddenly started colonizing much better. It showed that the leaked compounds themselves, transferred in the medium, were providing the growth boost.
Right, confirms the nutrients are in the exudate. But the final step proving it's the glenn they're eating, not just something else that leaked out too.
Yep, final piece of the puzzle. They made one more set of mutants, the GLN non-eaters. These were bacterial strains specifically engineered so they couldn't use GLN as their primary food source, couldn't metabolize it for carbon and nitrogen. And predictably, these non-eaters showed way less growth, significantly reduced proliferation, specifically on those leaky mutant roots. So that confirms it. GLN isn't just the signal drawing them in, it's also the main high-energy meal, fueling the population boom right at those leakage sites.
This is fascinating. It means that even on a perfectly healthy, normal root, we should expect to see these little clusters of microbial activity wherever there's a temporary natural break in that Casparian strip. So where do these natural leaks actually happen on a typical root?
We now understand there are basically 3 main types of transient natural hotspots on healthy roots. First, there's the area right near the growing root tip, before the Casparian strip is fully formed and sealed everything up. That's the pre-CS zone. Second, you can just get occasional spots where the CS is naturally a bit weaker or damaged just through normal processes, CS defective areas. And third, a really important one, the sites where lateral roots emerge, or LRE sites.
LRE, that's where a new side root literally pushes its way out from the main root.
Yeah, I can see how that would cause some temporary damage to the seal. Precisely. It has to physically break through the endodermis, creating a temporary breach, a short-lived little window of opportunity with leaking gill at end. And they visualize this beautifully. They used a special reporter strain of bacteria when engineered to glow green only when it was actively metabolizing GN. They saw this green glow pop up specifically around those emerging lateral roots, showing the bacteria were feasting right there. And crucially, that glow, that feeding zone, was tiny. It dropped off really quickly, just a small distance away. We're talking tens of micrometers. It shows how localized these nutrient differences really are. The whole structure of the root microbial community is being built, at least in part, on these incredibly small-scale transient leaks.
Wow. Okay, so this leakage idea forces us to kind of rethink how roots release stuff, doesn't it? We used to think mainly about like active secretion through special transporters or maybe shedding whole cells. Where does this gillen leakage fit in?
Yeah, it really adds a third pathway for exudation that wasn't fully appreciated before. It's the uncontrolled release of these small, valuable molecules like amino acids directly from the plant's internal transport system, the vasculature. And it happens specifically because of a failure in a physical barrier, the Casparian strip. It's not the plant deliberately feeding microbes here, it's the plant essentially failing to keep its high-pressure internal system completely sealed.
But hang on, if Glenio is so important for shipping nitrogen up to the leaves, doesn't leaking potentially loads of it cause problems for the plant itself, even before we worry about the bacteria? And what are the consequences of chronically overfeeding the microbes right at your doorstep, like those leaky mutants do?
You've put your finger right on the core trade-off. Yes, an intact barrier is vital for the plant's own nutrient efficiency. Losing gland is costly. But the failure also has big implications for plant health and defense. What they found in those leaky barrier mutants was quite interesting. Because there was this constant leak, they had this chronic overgrowth of bacteria, even just normal, generally harmless commensal bacteria. And this constant crowd triggered a kind of low-level but chronic immune stimulation in the plant. The plant's defense systems were always slightly switched on.
So the plant is basically living in a state of permanent low-grade alert?
Exactly.
Yeah.
That'd have a cost. It drains resources. While the leaky mutant plants seemed okay dealing with just the regular friendly microbes, they became hypersensitive when challenged with opportunistic pathogens, bacteria, that aren't always harmful but can cause disease if conditions are right. These pathogens absolutely thrived on all that extra ghreline leaking out, and they cause much worse growth problems for the leaky mutants compared to the normal plants. So the big takeaway here is that the endodermal barrier, the Casparian strip, it's not just about managing internal nutrient flow. It's also critical for limiting how much food you offer up, as a way to manage the microbial crowd size and composition, keeping potentially harmful members in check.
Right, so it's this really dynamic picture. You have these short-lived, super valuable feeding spots, these metabolic niches driven by developmental events like lateral root growth, causing temporary leaks. And these hotspots are essentially seeding the root community, attracting the first wave of microbes.
That's a great way to put it. It's a beautifully simple yet fundamental mechanism. Keep the dam intact, or you inadvertently feed the flood downstream.
So recapping this deep dive, the integrity of that Casparian strip is the gatekeeper. When it's breached, even temporarily, glutamine leaks out from the plant's internal transport system. And this leaked galen acts as both a powerful magnet, a chemotactic signal, and the main course, a carbon and nitrogen source, defining these hotspots where microbial life congregates on the root. It's like the blueprint for the rhizosphere architecture.
Absolutely. And what's really exciting is that this probably isn't just an Arabidopsis story. Glutamine, or very similar amino acids like asparagine, they are the standard nitrogen transport molecules in the xylem sap of tons of different plants, including major crops we rely on. So this mechanism of leak-driven colonization is likely incredibly widespread. It's probably happening in fields and forests everywhere.
Which, of course, leads to a really interesting thought for you, our listeners, to chew on. If these endodermal leaks, these transient hotspots, are such key control points for microbial access, what could that mean for the future of agriculture? We know things like environmental stress or different growth stages can change what's in the plant's xylem sap, maybe changing the gland concentration. Could we perhaps learn to strategically manage a crop plant's endodermal integrity? Maybe influence when and where lateral roots form to control these leakage events? Could we use this knowledge to selectively encourage beneficial microbes, the ones that help with stress tolerance or nutrient uptake, to colonize the root first? What new doors does this open for, say, microbiome-informed breeding in the coming years? Something to think about.